Hello, dear students. I, Dr. Rawal, your original guru for dental anatomy and dental histology, is here with you to discuss the questions that were asked in Rajasthan MO paper 2024, right? For subject DADH. So let's start with our first question. Which of the following types of fissures are most prone to caries? Is it inverted Y type? Is it IK type? Is it I type? Or is it V type? Actually, the morphology of the fissures mainly corresponds to the susceptibility of the caries. So here V and U fissures are wide and shallow type of fissures. So they are less prone to caries. While IK, inverted Y and I are deep and narrow fissures. Deep and narrow fissures. So as they're deep, they're more susceptible to caries as they're inaccessible based on their morphology. They are not properly cleaned. Proper oral hygiene cannot be maintained. Accumulation of plaque, accumulation of bacteria, resulting in more susceptible dental caries. So these are pits and fissures, and according to Nagano's classification, there are 5 types of fissures, mainly V type, U type, K type, and inverted Y and I type. So as we have discussed, V and U type are mainly shallow and wide fissures. So they are less susceptible to dental caries. While these are deep fissures, as we can see in this diagram, and I type, they are narrow and deep fissures. So they are more susceptible. And amongst the 3, most susceptible is inverted Y type because of its shape mainly. So the correct answer here is for inverted Y type. So we have discussed about the 5 types of fissures here. V and U shallow, and the rest of them are deep fissures. Formation of dental lamina is initiated by. Now, mainly the questions were from dental histology, right? And only from one topic, that is tooth development. And only remaining one was from salivary gland. Most of the topics were from topic of dental histology, that is tooth development. Formation of dental lamina is initiated by. Is it neural crest cells? Is it forebrain? Is it odontoblast? Or is it rest case pouch? Now, we have discussed in our recorded lectures as well as live lectures that all the dental tissues except enamel are formed by neural crest cells by the interaction of epithelium and ectomesenchyme. There is initiation of tooth development as we have discussed, right? So mainly tooth development is regulated by interaction between epithelium and mesenchyme. Neural crest cells form what? Mesenchyme, connective tissue portion, ectomesenchymal portion. These cells induce the send signals to the oral cells of oral epithelium which proliferate and lead to the formation of thickened epithelial band which separates into 2 parts, that is dental lamina and vestibular lamina. So mainly dental lamina is formed by neural crest cells, not directly by neural crest cells. It is formed by the interaction between epithelium and connective tissue. As connective tissue is formed by neural crest cells, this neural crest cells, this is our epithelial portion, this is our ectomesenchymal portion, right? So this ectomesenchymal cells are mainly neural crest cells. They send signal to the epithelium, leading to the proliferation of the epithelial cells, forming dental lamina. So indirectly, what is involved in the formation of dental lamina is neural crest cells. Crest cells. So this is the connective tissue portion and this is the epithelial portion. And this connective tissue sending signals to the epithelium, and the interaction between both of them leads to the formation of tooth, initiation of tooth formation. So amongst the option given here, the correct answer will be what? Neural crest cells. As enamel is an ectodermal structure, it is mainly formed by epithelial portion. Ectoderm. Rest are formed from ectomesenchyme, which is formed by neural crest cells. So, neural crest cells mainly induce the cells of oral epithelium to form dental lamina by sending signals. This is mainly reciprocal induction. The predominant immunoglobulin in saliva is it IgG, IgE, IgD or IgA? The correct answer here is IgA. IgA acts as a first line of defense against pathogens. It is present in highest concentration in saliva. So the correct answer here is IgA. This is the only question which was other than tooth development asked from dental histology. Remaining all the questions were from topic tooth development. Ectodermal cells in the tooth bud are responsible for. So we have discussed, right? Ectoderm, enamel is formed from ectodermal portion. Tooth bud comprises of three parts: enamel organ, dental papilla and dental sac. So enamel organs forms what? Enamel. Enamel organ is basically ectodermal structure. So ectodermal cells are involved in formation of what? Shape and crown of tooth, as enamel determines the crown of the tooth. So shape and crown of the tooth. Rest are formation of medial cementum and pulp are mainly formed from dental papilla and dental sac. So pulp is formed from what? Dental papilla. Right? PDL and cementum are formed from dental sac. They are formed from dental sac. And the remaining answer here is determination of shape and crown of the tooth. And this is the correct answer here. Rest of them are not the correct answer. As enamel is formed from what? Enamel organ. And enamel organ is basically what? It is an ectodermal structure. It is an ectodermal structure. So the correct answer is that ectodermal cells mainly determine the shape and crown of the tooth as they are involved in the formation of enamel. Right? So in this diagram, we can easily appreciate the ectomesenchymal structure. Here, we can see this is ectomesenchyme. This is ectomesenchyme, and this is the epithelial portion. It is the epithelial portion. So rest of the tissues are mainly formed from the ectomesenchyme, and only enamel will be formed from enamel organ. For the dental lamina question also, you can see this is our dental lamina cost, and this is the ectomesenchymal portion. This ectomesenchymal portion will send signal to the dental lamina, leading to the proliferation of the cells and forming enamel organ. So enamel organ here, this is enamel organ. It forms what? Enamel. Tooth bud mainly comprises of three parts: enamel organ, dental papilla, and dental sac. Enamel organ is mainly ectodermal structure and it is involved in determining the shape and crown of the tooth as it forms enamel. Now coming to our next question. Membrana preformativa is seen in. Is this seen in cap stage, advanced bell stage, bud stage, or bell stage? It is seen in early bell stage, not in advanced bell stage. Why? It is seen in early bell stage, not advanced bell stage. Why? Because advanced bell stage, you can see formation of hard tissue. Hard tissue is basically dentine and enamel. What is formed first? Dentine or enamel? Dentine. So here it is asked, membrana preformativa is seen. It is mainly seen before hard tissue formation. As it is seen before hard tissue formation, so hard tissue formation is basically involved in advanced bell stage. So it cannot be seen in advanced bell stage. It is mainly seen in early bell stage. What is membrana preformativa? It is a basement membrane between inner enamel epithelium and dental papilla. Right. So here you can see this is dental papilla and these cells are inner enamel epithelial cells. And between them we have layer that is membrana preformativa, the basement membrane type of structure separating the inner enamel epithelium from the dental papilla. So this is dental papilla and above it there are inner enamel epithelial cells. You can easily appreciate the inner enamel epithelial cells here. These are inner enamel epithelial cells and the green portion you are seeing that will be the basement membrane separating the inner enamel epithelium from the dental papilla. And this structure is membrana preformativa before the formation of dentine. So remember, formation of dentine, any hard structure starts in advanced bell stage. It forms a blueprint for the shape of the crown. Now coming to our next question. First appearance of tooth formation occurs in, so this is very basic question, it occurs in 6th week of intrauterine life or 4th month? No, not 4th month. It is 6th week of intrauterine life. Why 6th week? Because at about 6th week, there is a rupture of buccopharyngeal membrane and there is the connection between the epithelium and the connective tissue. Again, the same point, interaction between the epithelium and the connective tissue leads to the initiation of the tooth development. So the answer here is 6th week. As at about 6th week, the main junction between the buccopharyngeal membrane that is ruptures and there is the connection between epithelium and the connective tissue, sending signals leading to the formation of tooth. So the correct answer here is 6th week of intrauterine life. Blood supply of ameloblast for most of its life cycle comes from, again this is question which is our last question and it is again from tooth development. So it comes from dental pulp, dental papilla, reduced enamel organ or dental sac. This is again an important question from tooth development. Which stage? Advanced bell stage. Here it is asked blood supply from ameloblast for most of its cycle. Initially, ameloblast cells get nourishment from dental papilla, as we had seen in early bell stage diagram. Remember, this is our early bell stage diagram. So initially, we can see it is receiving nourishment from dental papilla. Which cells from enamel? Inner enamel epithelial cells form enamel. So this inner enamel epithelial cells differentiate in ameloblast and form enamel. So this inner enamel epithelial cells are getting nourishment from dental papilla initially during early bell stage. But once dentine is formed, hard tissue is formed here. If hard tissue is formed here, what will happen? The blood supply will be cut off from the dental papilla. This diagram we can appreciate. Here we can see dentine formation. This is dentine. Dentine formation we can see. So once dentine is formed, blood supply from dental papilla will be cut off. There will be no nourishment from dental papilla. No nourishment from dental papilla. So now what? For metabolic activity, they need nourishment, nutrition. From where will they get nourishment? Inner enamel epithelial cells. Otherwise, they won't be able to convert into ameloblast and enamel won't be formed. So now they will receive nourishment from dental sac. This is dental sac. Dental sac. They will receive nourishment from dental sac. So as the nourishment from dental papilla is cut off, once dentin is formed, they will get nourishment from dental sac. As a result of which the outer enamel epithelium is laid down in folds and the direct nourishment can reach the inner enamel epithelial cells. So basically, this from dental sac. And then inner enamel epithelium. First, dentin is formed. After that, enamel is laid down. So without inner enamel epithelial cells, signals won't be sent to dental papilla and it will not differentiate into dentin. Once dentin is formed, it will send signals to inner enamel epithelium cells and they will form enamel. This is known as reciprocal induction. Formation of dentin and enamel is mainly interdependent. Without formation of dentin, enamel cannot be formed, and without inner enamel epithelial cells, dentin cannot be formed because they send signals to the connective tissue cells, that is dental papilla, and form dentin. So here mainly what was our question? That blood supply of ameloblast is from dental sac. Because once dentin is formed, nutritional supply will be cut off from dental papilla. So rest of them are the correct option. So there were mainly 7 questions. We have discussed all the questions and most of the questions were from tooth development, which is very important topic.